Good afternoon, everyone. Thank you for joining us today for this special earthinar as we continue to mark the launch of the new Center to Advance Toxicology and Chemical Hazard Assessment, also known as CATCHA. I'm pleased to introduce to you Dr. Joshua Robinson, who is an associate professor at UCSF in the Department of Obstetrics, Gynecology, and Reproductive Sciences. He is also the PI of the CATCHA Center, and he's one of Earth Center's consultants on our Integrated Health Sciences Facility Core, providing consultation and chemical selection, measurements, and model systems. Let Josh take it from here as he introduces today's special speaker and gives you a little overview on the CATCHA Center. Josh?
Oh, thanks, Alana. Thank you for that intro. So as Alana mentioned, we're excited to co-host the seminar with the UCSF Earth Center. Again, we are establishing this center that's focused on advancing toxicology and chemical hazard assessment with the mission to develop, advance, and establish new science strategies for chemical hazard identification and risk assessment. Really interested in gathering individuals who are interested in a variety of different areas, including in vitro and silico modeling, translational approaches, and hazard identification and risk assessment. Next slide, Alana, please. And with that, we have several ongoing activities, including co-hosting Earth Center webinars like today's, which I think really shows wonderful research being done in this area by Dr. Everson, who I will introduce shortly. Also, we have another webinar that will come up in February that we're very excited about by Dr. Vera Cologne and Nguyen at Berkeley. And again, we want to grow this center in a variety of different ways, including all that is to find funding opportunities. We'll be announcing some pilot project funding opportunities in the next couple months, as well as thinking of ways that we can do team science together to position for upcoming grant applications. So please reach out and either email myself or Ariel. So with that, I'd like to introduce you to Dr. Josh Everson, who is our guest presenter today. He is a postdoc in the lab of Dr. Johan Everhart at the University of Texas at Austin, where he's studying the underlying gene-environment interactions that lead to craniofacial defects using advanced genetic and molecular approaches in zebrafish as a model. His training includes a PhD in molecular and environmental toxicology from the University of Wisconsin-Madison, where he worked with Dr. Rob Lipinski, and additional experience working in Dr. Kathy Solik's lab at the University of North Carolina at Chapel Hill. He recently was awarded the Society of Toxicology and Society of Birth Defects Research and Prevention Edward Carney Trainee Award and the NIH K99 Independence Award due to his tremendous early success. And he is currently pursuing academic faculty positions to establish his own research program in developmental biology and toxicology. Today, he'll be sharing some of his work in a presentation titled A Universe of Toxicants, Universally Undertested, Higher Throughput Approaches to Birth Defect Detection in Fluorescent Zebrafish. Thank you, Josh.
Yeah. All right. Good morning, everyone, and thanks for being here. One second. There you go. My name is Josh Everson. Like Josh just said, I'm a postdoc at UT Austin. Today I'll be telling you about my work developing a new platform for the high throughput detection of birth defects using zebrafish. So as an overview, I'm first going to tell you about the chemical testing problem, that there are tens of thousands of chemicals in the environment, of which most are undertested. And this is in large part due to common testing methods like mouse, rat, and rabbit developmental toxicity assays being far too slow. So therefore, we need to develop faster methods for chemical testing. And as I'll tell you, zebrafish have several advantages that enable early high throughput chemical testing and birth defect detection. So today, I'm going to tell you about my development and validation of this assay from the ground up. And then I'll end with how I plan on using this assay in my K99 work for the rapid testing and prediction of gene environment and chemical interactions. So why is this needed? Well, at least 3% of babies born each year have a birth defect. However, for the vast majority, up to 80%, we don't know the cause. And why is this? Well, we think many birth defects result from complex interactions between multiple factors, both genetic and environmental, which has made testing slower and also identification of specific causes difficult. Okay, so when it comes to the environment, we know very little about what toxicants may be contributing to birth defects. There are over 80,000 EPA registered chemicals, with more than 700 new chemicals entering the marketplace each year, 140,000 chemicals that have been commercialized, and 85 million that have been synthesized, which humans could one day be exposed to. However, the safety of the vast majority of these chemicals is completely unknown. So of the 80,000 EPA registered chemicals, only about 1,000 have been well studied with in vivo assays, and only about 10 to 20,000 have been studied at all for any endpoint, which is mostly cell-based and in vitro systems. So how did this happen? Well, the problem is that traditional testing, like I said, is too slow and often very expensive. So the most common EPA protocol for developmental toxicity assessment is maternal exposure in mice, rats, and rabbits. But these organisms have a slow gestation lasting 18 to 30 days. And with exposures targeting organogenesis, those exposures have to span 2 weeks. And lastly, regardless of the model organism, traditional testing endpoints like histology and morphological phenotyping are very time intensive. So in some, these models are just simply too slow to get through the number of chemicals that we need to get through. So in response, the EPA partnered with the NIH to form an initiative that's aimed at updating toxicity testing for the 21st century. And they have a particular focus, similar to this center, on using new platforms for more rapid chemical testing. So this initiative has produced 2 large data sets that I'd like to mention today. The first is an extremely impressive giant data set of over 10,000 chemicals for 70 different cell-based assays. So the EPA intentionally focused on these in vitro assays to maximize throughput while also providing mechanistic data, which makes a ton of sense for many toxicological endpoints. But for something like development that's very complex, these in vitro systems aren't likely going to capture that complexity. So a second data set contains developmental toxicity data for over 1,000 chemicals, which is still very impressive. And it identified 178 chemicals as being craniofacial toxicants. This work was in vivo. However, the endpoints are morphological and therefore lack any mechanistic data. And this mechanistic data is crucial, as it's what we need if we hope to predict low dose effects, mixture effects, and also to predict gene environment interactions. So one of the goals of my postdoc was to try to bridge the difference between these 2 strategies to develop a higher throughput method for in vivo developmental toxicity assessment that would also be capable of providing mechanistic insight to predict these gene environment and gene chemical interactions. So complicating matters further, like I said, many human birth defects are thought to result from multifactorial gene environment interactions, where predisposing mutations interact with environmental exposures to cause birth defects. And one study that examined the most common craniofacial malformation, that being orifacial clefts, like cleft lip and cleft palate, it identified 363 genes that were associated with that disease, meaning that there's at least this many genes that regulate craniofacial development, which could conceivably act in gene environment interactions. And on the other hand, I just mentioned that the largest in vivo screen to date, which was 1,000 chemicals, identified 178, or just about 10% of the chemicals that they tested, that caused craniofacial malformations by these unknown mechanisms. And gene environment interactions are critical from the perspective of risk assessment, because a sensitizing mutation can make a dose that's otherwise not toxic and considered safe into one that could cause birth defects or other toxicity. And this concept is also true for mixtures of chemicals, which interact either additively or synergistically. So what does this all mean? Well, unless we develop better and faster ways to screen chemicals and to predict interactions, we're going to have a lot of very overworked toxicologists. Okay, so a major goal of my postdoc was to move into a new model organism that would enable higher throughput analysis. And for several reasons, I thought that the zebrafish looked perfect. Because despite the common idiom, breed like rabbits, we should really say spawn like fish. Because while a pair of rabbits, or mice for that matter, can produce up to a dozen offspring per litter, and as mentioned, will take 20 to 30 days for gestation. A pair of zebrafish can produce hundreds of embryos, and they can be rebred every week. So this means a bounty of embryos that rapidly develop in only 5 days. And in addition, the embryos develop externally, which is ideal for these tightly controlled chemical exposures, and also critical for the assay that I'm about to tell you about. Zebrafish embryos are optically clear at early stages of development. So together, I thought that these attributes would enable high throughput analysis of live embryos using a fluorescent plate reader based on the following hypothesis. So something that we know is that a common feature of many, if not all or most craniofacial birth defects are changes of the cells that form the face. And there are transgenic zebrafish lines where these facial precursor cells are endogenously labeled fluorescently, an example of which is shown right here on the right. So my hypothesis was that if early in the adverse outcome pathway of facial malformations, there are changes in these cells that could be captured using a fluorescent plate reader, then defects could be detected earlier and faster, providing this much needed throughput and also providing a live in vivo assay for developmental toxicity testing. So what are these cells? Much of the facial tissues are derived from a transient developmental cell population called the neural crest cells. Shown here are scanning electron micrographs of mouse development over time that I took during grad school, with the equivalent staging in human, mouse, and zebrafish. So these neural crest cells are first born in the neural plate very early in development right here. And then they migrate out of the developing brain to populate the future face, which are, you can see here these facial growth centers, which are these prominences on the front of the embryo. So if we chop into one of these facial prominences, you'll see that they are comprised and filled in with mesenchymal cells, which is what the M here stands for, which are derived from the neural crest. And you've already seen these same cells on the previous slide in a 24-hour zebrafish embryo labeled with a fluorescent marker. So these cells will then respond to highly conserved developmental signals from the surrounding tissues that tell them to differentiate into the various bones and cartilages of the face. And you can see this with the highly developed faces of these mouse embryos here, or in this stained zebrafish embryo at 5 days old, where the cartilage has been stained blue. And this series of images demonstrate a couple strengths that I mentioned of zebrafish. The first is their rapid development. So take note of the units here. You'll see that what takes days or weeks in humans and mouse occur in just hours in a zebrafish. And in addition to showcasing another strength of zebrafish, one that I've leveraged heavily for this assay that I'm about to tell you about, which is their external development and also their optical clarity. So unlike the zebra or the mouse embryo, which I showed you, we have to euthanize, cut into, and to physically view the neural crest cells. With a zebrafish, which is optically clear and with transgenic lines where fluorescent markers can be expressed, we can see these cells inside the embryo in vivo in a live animal. So I target my drug exposures for the duration of this talk to early development, trying to cover these early stages of neural crest development. And notably, at the time of the start of drug exposure, the embryo, whether it's a human or a mouse, is so small that it can fit on the nose of Abe Lincoln, and also critical from the perspective of birth defect prevention. At these stages of development, most people will not yet even know that they're pregnant. So following drug administration, which I just talked about in A, I then removed the embryos from their shells and put them into a 96-well plate, 1 embryo per well. And as I mentioned, these embryos harbor a fluorescent tag, meaning that fluorescence can then be measured using a plate reader, which is shown here in C. And then importantly, since this is a live in vivo assay, the embryos can then be developed the additional 4 days for their facial skeletons to form, at which point normally defects could be measured, which is a major strength of the assay because it allows us to directly pairwise compare whether fluorescence early in development led to the expected changes in facial morphology. And then we can test whether these fluorescent changes are actually predictive of later defects. So what is the plate reader doing? For each well, so I'll walk through for a single well. For each well, the plate reader first captures a bright field and then a GFP image. And it then takes a 5x5 array where it scans for the GFP expression or whatever fluorophore you're targeting in 5 specific regions, providing 5 readings per well. So this analysis, I was focused on capturing craniofacial defects. So I then thresholded these 25 readings to capture only those where the marker fluorescence is the brightest, which for in this case was above the 3rd quartile. And for this marker represents the head. And then these readings are then, that pass threshold, are then summed to give a single value for each embryo. And of course, since this is in a 96-hole plate, this can be repeated automatically and in high throughput to gather data on 96 embryos. So for example, a typical plate setup, you might have 4 treatment groups, control, and 3 like low, moderate, high doses. And it can capture all of this in about 20 minutes. And in the end, it delivers quantitative well reads for each embryo across treatment groups, which is very easily analyzable using statistical statistics like ANOVA to determine dose-dependent effects. So to first assess the ability of this assay to detect neurocrest or craniofacial defects, I turned to one of the most common environmental teratogens and one that I've studied extensively in the lab and outside of the lab, which is alcohol. Previous work by myself and others has shown that doses of alcohol higher than 1% in the media cause craniofacial malformations in zebrafish. So I therefore exposed embryos to either 1% alcohol or greater and measured changes in their marker fluorescence. So A through D here show side views of representative embryos. And to orient you, this is the eye right here, and the white cells are the cells that express the marker gene. And on the right is quantification of each embryo's cumulative read, with each circle representing a single embryo, and then the mean SEM and significance by ANOVA shown for each group. And consistent with the previous work and providing an exciting proof of principle, I found that alcohol doses greater than 1%, but not 1% alcohol itself, caused significant reductions in the reporter's fluorescence. This suggests that early reductions in reported fluorescence can indeed predict later defects. But like I said, we can actually test this directly because this is a live assay. So we can leverage that strength in order to directly examine whether the fluorescent readings at 1 day correlate with craniofacial defects later, with the hypothesis being that those fish with the lower fluorescence readings at 1 day will be the ones will end up with smaller facial elements, and vice versa. So to accomplish this, I worked with an undergraduate researcher here at UT to perform staining and linear measurements on the craniofacial skeletons of 5-day-old control and alcohol-exposed embryos whose fluorescence had been measured at 1 day old. So as shown here are data for 2 of the endpoints that he measured, the neurocranium, which is essentially the entire length of the skull, and then the trabecula, which is this small structure in the middle here, labeled TL, which is analogous to the roof of the oral cavity and the palate in mammals. And which important for this, fate mapping has shown is almost entirely comprised of neurocrest cells, versus the neurocranium is mostly neurocrest derived, but this posterior part is not neurocrest derived. So each graph shows linear measurements for the facial element, with again, each dot representing an embryo, and the mean SEM and significance by ANOVA being shown. And consistent with the previous work, this analysis again showed that only doses of alcohol that were greater than 1% caused significant reductions, and that the 1% dose does not. So now that we had 2 quantitative endpoints for each fish, we could easily and directly correlate these values in a pairwise analysis to test whether reduced fluorescence early correlates with morphology later. And to my excitement, that's exactly what we found. So shown here are scatter plots for those 2 facial elements, the neurocranium and the trabecula, with the fluorescence shown on the y-axis and the x-axis showing the measurement, the length measurement. And what we found is a significant correlation between marker correlation and the length of each craniofacial element, which you can see here with the Pearson's coefficient p-value. And interestingly, and consistent with the hypothesis that these early cell changes will predict later outcomes, the strongest correlation was found for the trabecula, which, like I said, is almost entirely comprised of the marker gene expressing neural crest cells. So together, this demonstrated that the plate reader can detect these pathogenic changes that predict later outcomes. And these changes occur early, before the face has even finished developing, which allows for much earlier analysis in gestation. So for example, for a zebrafish, you can analyze 4 times sooner. For a mouse, this would be even more, like 10 to 20 times sooner than you could. And in addition, the assay itself and the analysis of the data it produces is very fast. It takes only about 20 minutes to scan 96 embryos, which for comparison, just to do the imaging of 96 embryos would take at least 5 times longer, at which point you still need to take linear measurements or do manual scoring. So therefore, conservatively, the down-Q analysis is at least 10 times faster, but likely much more than traditional staining and histology. So now that we convinced ourselves that the assay does work, we moved on to more interesting test scenarios. So as I mentioned, we think that most birth defects are the result of multifactorial interactions, including co-exposures. So I wondered whether the plate reader could capture chemical interaction and predict mechanisms. But first, I want to give some background on the chemicals that we'll be looking at. So while I was in grad school, I spent a large portion of my time examining a common pesticide additive that's called piperonal butoxide, or PBO. So developed in the 1940s, this chemical can be found in thousands of household products, including lice shampoos and indoor bug bombs. And being a synergist and not an active pesticide, PBO has received relatively little regulatory attention. And somewhat terrifyingly, a 2012 study showed that this chemical can inhibit the hedgehog signaling pathway by the same mechanism as the classic one-eyed sheep-inducing teratogen cyclopamine, which was discovered after pregnant sheep ate this plant, which I saw and took a picture of in Utah at an SDB, which contains cyclopamine. And this plant is called the California corn lily, so I'm growing all over the place there. That's how it works. So we showed that PBO is only 4 times less potent at blocking the hedgehog pathway than cyclopamine, which you can see by comparing the EC50 values here. And just for note, vismodegib is a positive control anti-cancer drug that's extremely potent, as you can see here. So based on this, I then developed dosing strategies and exposed pregnant mice to piperonal butoxide at the same time points I've been showing you so far, so gastrulation, and found that PBO can cause severe sonic hedgehog associated brain malformation called holoprosencephaly, which has co-occurring associated midfacial defects. And I found that this can happen at doses as low as 66 mg per kg. Which you can see with what I took here is specifically were linear measurements of the interocular distance, so a midfacial measurement, and then the upper lip length, which becomes seemingly longer, but it's due to the loss of this midfacial tissue. And note that the previous EPA accepted low effect level for PBO was 1200 mg per kg, which was determined in an untargeted analysis that predated the discovery of PBO's ability to block the hedgehog pathway. So it really shows the importance of doing these targeted developmental toxicity screens where we have a mechanism of action in mind. So in addition, I showed that mice with single allele mutations in sonic hedgehog are sensitized to PBO-induced malformation, which you can again see from the reduced interocular distance and increased upper lip length of the heterozygous animals compared to their wild-type siblings exposed to the same dose. So this demonstrates that gene-environment interactions can further raise the developmental risk of piperonal butoxide for these sensitized populations, and also suggest that PBO is working through the hedgehog pathway to cause these defects. And then similarly, while I was working at UNC as between undergraduate and graduate school, I helped show that these same sonic hedgehog heterozygous mice are also sensitized to alcohol-induced facial defect following exposure at the same time in development gastrulation. And similar to PBO, this suggests that alcohol, at least in part, elicits its toxicity, whether direct or indirectly, through modulation of the hedgehog signaling pathway. So when I started my postdoc, one of the first things I wanted to do was test co-exposure to alcohol and piperonal butoxide, something that's much harder to do in something like mice compared to zebrafish. So what I did was I exposed zebrafish embryos to either PBO, ethanol, or both chemicals from gastrulation to pharyngeal stage. And I found that PBO could indeed interact to cause defects. So you can see that by comparing these last 2 bars. So the second to last bar shows animal or the effect of piperonal butoxide and ethanol. And then if you compare that to this bar on the right, that's piperonal butoxide only, or this bar on the far left is alcohol only. So you can see that the effect of the co-exposure was much more severe than the effect of either chemical on their own. So this demonstrates a multifactorial chemical interaction centering on the hedgehog pathway. And I thought that this would be the perfect positive control to test the plate reader's ability to capture mixture interactions between two toxicants. So I exposed zebrafish embryos to either a dose of PBO or a dose of alcohol that on their own caused few malformations, and that was 1% alcohol or 25 micromolar piperonal butoxide, or the two chemicals on their own. And consistent with the previous work, PBO ethanol co-exposure, but not the single exposures, caused a significant reduction in fluorescence. And you can see that these single exposures do have an effect. It's just not significant, showing that the assay is sensitive enough to capture changes that the embryo is able to ultimately tolerate, and then also changes which the embryo cannot tolerate. So this is very exciting because it really opens the door for faster screens. However, directly testing interactions between even just the 187 chemicals identified by Tangway's group remains an immense number of experiments, and that's even before we start considering gene-environment interactions. So I wondered, what if we could do it better yet? So if these chemicals cause malformations by perturbing developmental signals, and given that we have transgenic lines that we can use to monitor the activity of these signaling pathways, I wondered if the plate reader could detect chemical-induced pathway perturbations, which we could then use to provide insights on their mechanism of action. And then based on the hypothesis that chemicals that cause similar pathway perturbations will interact the most strongly, then perhaps this assay could be used to prioritize and predict chemical interactions. And then finally, since it will provide insights on each toxicant mechanism of action, I wondered if it could be used to predict genes in those pathways, which the chemical would be predicted to interact with in gene-environment interactions. So I started this adventure with my favorite signaling pathway, the hedgehog pathway, because it's a critical regulator of brain-phase development, and also, as my data shows and others have shown, it's really a gold standard for gene-environment interactions in birth defects. So I showed you earlier that two common environmental teratogens, piperonal butoxide and alcohol, can interact to cause defects, and that each chemical's toxic effects are exacerbated in mice that have sonic hedgehog mutations. So I first tested the conservation of this gene-environment interaction. So embryos that were either wild type or heterozygous for sonic hedgehog were exposed to increasing doses of either alcohol or piperonal butoxide. And in both cases, as you can see, malformations were more severe in the heterozygous animals compared to their wild type siblings, demonstrating conservation of this gene environment interaction, and again, suggesting that these chemicals in part elicit their effects through disruption of the hedgehog. So now that we knew that these chemicals could interact and likely through the hedgehog pathway, I thought they could be used to work backwards to provide a proof of principle that the plate reader could capture early pathway-specific disruptions, and in doing so, be able to predict chemicals that would be most likely to interact. So first, as a positive control, I tested just the ability of the plate reader to capture cellular and molecular disruption. So I've already shown some cellular data, but now we're going to move on to molecular disruption. Early in the pathogenesis of birth defects, in this case, birth defects caused by exposure to a hedgehog pathway inhibitor. And so I exposed embryos to a sonic hedgehog pathway inhibitor, which I'll just call BMS here, and found that doses at or above 3.125 micromolar caused significant malformations. So that's shown here, sorry. And when I tested these same doses earlier using the plate reader and the neural crest cell marker, I found that the same dose, 3.125, was a low effect level for the reduced fluorescence. So now finally getting to the crux to test if we could use this to gain these mechanistic insights, I utilized a different transgenic line. In this case, it's a hedgehog pathway readout, which has multiple GLE binding sites, which are the downstream effector gene transcription factor. So these binding sites get bound by GLE and it activates expression of fluorescent protein M-cherry in this case, which allows for real-time monitoring of the hedgehog pathway in live. And what I found is that significant reductions in the hedgehog pathway-specific fluorescence occurred at these higher doses, but very interestingly, the lowest dose that we saw a significant reduction was also the same dose, 3 micromolar. So this really provides a strong proof of principle that the plate reader can be used to capture pathway-specific perturbations in real time during the early pathogenesis of birth defects. So then finally, I return to our prototypical interactor chemicals, the pesticide synergistic propionyl butoxide and alcohol, to test whether, as predicted by my hypothesis, each chemical would cause detectable losses in hedgehog-specific fluorescence. So I exposed zebrafish that contained this construct to increasing doses of PBO or alcohol. And I found that both PBO and alcohol caused reductions in this fluorescent marker, which suggests that they work by similar mechanisms. And if we were to imagine that this pathway-specific assay was done first before the cell-specific or the staining of the faces to screen chemicals that have similar perturbations, then this would have predicted that these chemicals would interact. So it provides proof of principle that using these pathway-specific perturbations, we might be able to predict chemicals that would interact that we'll then validate later. So going forward, I'm expanding these studies to other craniofacial signaling pathways and other test chemicals. So I've developed standardized dosing protocols for inhibitors of 6 other signaling pathways, in addition to the hedgehog pathway that I showed you today. And each of these inhibitor standards will be paired with a pathway-specific readout. So these are the pathways, and each of these has a pathway-specific transgenic line that I have in hand that I will test in order to determine the signature of inhibition of each pathway. And then following that, I will then repeat those studies, but using these 13 environmental test chemicals from the Tangway data set in order to find chemicals that cause similar perturbation, with the concept being that those chemicals will be the most likely to interact with each other based on having similar mechanisms. And then in parallel with this, so I'm not talking about it today, I'm also using a transcriptomic approach to examine these same 6 pathway inhibitors and 13 environmental chemicals, which follows a similar logic to predict interactions, that chemicals that cause similar transcriptomic changes, meaning they impinge on similar genetic networks, will be those that are the most likely to interact and to interact the most strongly. But in both cases, these putative interactors will then be tested in high throughput using the NeuroCrest marker plate reader assay. So bringing this all full circle, I've shown you today that there are myriad chemicals and genes that may interact to cause craniofacial birth defects. And we know very little about these interactions, in large part due to tens of thousands of chemicals being understudied and uncharacterized, which itself is in large part due to the low throughput of our current testing methods. However, with the power of zebrafish transgenics, by using cell markers for the early detection of birth defects, or using pathway-specific readouts and transcriptomics to predict mechanisms of action, we can more rapidly characterize these developmental hazards, characterize interactions, and ultimately protect embryos and prevent birth defects. And with that, I'd like to finish by thanking my current lab and advisor, Dr. Johanna Everhart, for the wonderful and supportive lab environment that allowed me to pursue this crazy pipe dream. And lastly, I'd like to thank the NIH for funding this work, specifically the NIHS for my K99, previous funding from the NIAAA and NIDCR for my F32. And then lastly, I'd like to thank all of you for your attention today and for the opportunity to come here and share your work, or share my work with you. And I'd love to answer any questions.
Wonderful presentation, Josh. I think it really captures several exciting elements of this field in terms of using the zebrafish for chemical screening. And I just love all the genetic technologies you've added to it to make it a more high throughput system. Would you like to provide the first question, Dr. Marcuccio?
Oh, yeah, I can ask a question. I just wanted to turn my screen on.
Oh, thank you. I do appreciate that.
Yeah. Hey, Josh, that was fantastic, man. That was really cool. Cool application. You know, a couple of things I thought of. What if the embryos have just a brain defect? Do you think you'd see that with the neural crest markers?
I think that was part of the rationale, was that even if you hit the brain, it would probably cause secondary effects to the neural crest. But yeah, I would like to also incorporate other markers such as SOX2.
Yeah, that's what I was wondering. If you could combine other things to hit at different tissues, and would that work? I mean, I wonder if you had different fluorescent plate readers, different wavelengths. You could look at the brain, the crest, endoderm, whatever, heart.
Right. So I have done neural crest marks. So I've done SOX17 and SOX10 for neural crest endoderm. I haven't yet done brain and face. The other one I'm trying to do is get these pathway readouts into single animals as well.
Yeah.
Talk between pathways.
Yeah, that's really cool. The other question I had is in the gene environment stuff with the hedgehog, the mouse mutants. Did you ever get really severe defects, or you just get like this, this narrowing of the face? I mean, did you ever see well, there's two questions really. So you see a narrowing of the face, but did you see any increase in the variation?
Increase in the variance.
In the variance?
Oh, right. Yeah, we typically do see increases in variance. Most of these exposures that we do. We did also in that study, the mouse one, we did look at the brains and show that they do have true whole prosencephaly based on the histology of the brain. So we do see the more severe ones, typically only at 600 or 1800 mg per kg in a wild type animal. But yeah, we can see basically exact phenocopy of what vismodegib did, that positive control hedgehog pathway inhibitor.
Cool. That was really great talk. Say hello to Johan.
I will. Thanks, Ralph.
Maybe I could ask a question. Oh, sorry, Susan, would you like to go for me?
Sure. Or I was curious, excuse me, that you talked about things we would expect. Have you screened any chemicals just for fun and found some new things?
I haven't yet. That is something I'd like to do. I was talking with Josh about the possibility of doing this. So they've got data sets of chemicals that people have been exposed to, which we don't know anything about, whether they might cause defects, which I think would be very interesting to look at. Yeah, so far I've focused on things that we would expect to validate the model, but I do hope to expand this into much more, into the unknown.
And I was thinking, you have an assay with an organism that lives in water. It would be cool to test water from different sources to see if you can actually reverse engineer what's going on with regard to teratogens in water.
Yeah, that's a great idea too. Yeah, we have talked about some with Johan. So there are data sets from similar to what you're saying with, I know there's a big group in Texas that has identified water toxicants. So that's something else we've talked about. I've mostly been focusing on these ones because my K99 reviewers all thought that 13 chemicals was already way too overambitious, or it was originally 50. So I feel like to expand beyond these before confirming anything with these is just too far. But I do hope to expand definitely to more unknowns. Yeah.
Thank you.
Yeah, thank you.
Josh, I'll jump in here. I mean, so just for the Earth Center is kind of a broad audience. It'd be nice to just discuss kind of the concordance of the zebrafish and the use of it as a model with rodents and humans. We've talked a lot about the Tangway data set and kind of narrowing down on chemicals to test in your system. How much of those 178 chemicals do we have rodent data to kind of support that they have that causes this phenotype or a similar phenotype in rodents, and and is there connections, I would assume, with some of these in humans as well?
That's a great question. I don't know how many of them have parallel studies done in mouse. That's something I should definitely look at least for these top ones that I did, because I prioritized based on potency of causing malformations.
And so kind of understanding
Yeah, so you might imagine those would be the ones that we might have data for, just considering that they're so toxic.
And I would assume that there's a high level of concordance. It would be interesting if there's particular chemicals that do not show the same phenotype, right? Because that would be very interesting to understand sort of mechanistically, where these differences lie, right? When we're thinking about kind of the applicability domain of your system.
Right. Yeah.
I had another part to this too. This is a question. So as you know, we do rat embryo, whole embryo culture, which captures, it's not as high throughput by any means, but as compared to the zebrafish, but we deal with, we do a lot of morphological scoring. I could see how we could implement some of the technologies you've had, and I'd be very excited to think about that. We get this push about, are we actually capturing a defect versus a delay? And I was curious how much your system kind of picks that up, knowing that you probably understand craniofacial biology a little better than I do. Is there any part of this in the system and some of the readouts where these embryos could eventually develop back to normal, or it's just in a slower progression, or is this clearly going to be a defect of the craniofacial region?
Right. Yeah, that's a really interesting question. So I would say that the kind of longitudinal analysis showing that the neural crest changes translate into actual smaller facial elements would show that they are persistent changes. But I would also say that, yeah, this question of delay is an important one. And I think that since it's a live assay, we could do a time course in order to maybe see early changes that resolve, or maybe early changes that get worse. This is also something I was really hoping to do with looking at multiple pathway markers at once, because you might be able to see, say, expose an embryo to alcohol and see that first maybe hedgehog changes and then FGF changes after that, like that kind of thing. You might be able to get some insights on kind of how these pathways are talking to each other.
Yeah, I see that as a really advantage of the zebrafish. Embryo culture, or rat embryo culture, it's a little difficult. It's a little more kind of time point based, right? So we can't see this longitudinal effect, which, I think there's a big advantage there. Maybe you could dive in a little, and maybe if it's speculative, but kind of understand the interplay between PBO and ethanol. So I do know ethanol, from my knowledge, disrupts retinoic acid. And I do believe there's a connection there with sonic hedgehog. Is that sort of the mechanism you believe is at play, or is there other parts to it?
I still don't know exactly how ethanol is disrupting the hedgehog pathway. So yeah, there's the retinoic acid part that could definitely be part of it. There's also a new, like Robert Krauss's new model is that it's actually disrupting nodal signaling first, and then we're seeing these changes in hedgehog later. Like I haven't looked at that yet. I do have a data set where I did co-exposure to alcohol or that BMS, hedgehog pathway inhibitor, and did transcriptomics on those, and then also did single exposures. But to be honest, I haven't had time to really dive into it yet to see what might be happening. But it's one of my, I've worked with alcohol and hedgehog for like over 10 years now, and it's kind of demoralizing that I don't have a better answer of how it might be acting. But basically, I think the going hypothesis is that alcohol is doing a lot, and likely the effects on the hedgehog pathway are indirect. But I will say that we know that if you expose to alcohol, you'll get reductions in pathway readouts, like say patched expression, or like I showed with that cleat binding site. So it is clearly affecting the pathway, but yet the question is, how direct is that? And that I don't know.
Thank you.
Yeah, thank you.
So I have one more question. There's a lot of maybe more junior members on the call as well. I think to appreciate essentially the setup of a type of system like this with the fluorescent readout, do you have any kind of quick thoughts about all the troubleshooting that went behind it, and kind of I think some of the nuances with some of these different assays. I think, yeah, again, they're just underappreciated. It's hours and days and months that are put into it to get to this place. Maybe you could talk a little about that, like positioning of the embryo, like the different steps to get into the 96 well plate. I don't know, kind of what were the biggest challenges with maybe setting this up?
Right. Yeah, I would say the biggest one is the position of the embryo. So I really wanted to be able to do this in embryos that I didn't decorionate, because that's the eggshell that they're in. Because it would be so much faster and easier to work with because they're protected when they're inside it too. But when they're in that chorion, they kind of flip around frequently. So I was getting really noisy results, and one of the biggest changes was just removing them from the chorion, and then they sit nicely on their side. So that was probably one of the big ones. Also anesthetizing the embryos so that they don't move when they get hit with the laser, because that's another one. Then they'll move and you'll get a reading in 2 different spots. So that was another obvious big change. And then one of the things I still need to return to is I originally wanted to have more than 1 embryo in each well, because you can have up to 3. And I thought that would really increase the power. But I kind of, in order to do these correlations, that's why I ended up going with 1 embryo per well. But ultimately, I think it would be cool to go back to doing maybe 2 or 3 per well to just really increase throughput.
Yeah, and you have such clean data now, so you can kind of see how far you could expand it, right?
Yeah, and then I guess one of the other big questions now when I'm thinking about other fluorophores is, and which is probably going to be a issue with the machine itself, like how specific the wavelengths is. But basically, you can shorten or narrow the spectrum that you're going to capture the light from in order to get, so that you can have like 2 fluorophores in the same fish, but then you're going to lose signal. So that's one of the other big things that I need to work out going forward, because I'd really like to be able to express multiple fluorophores. And then I guess another related to that is the, I forgot the term for it, but how much light these fluorophores produce is also a big factor. So like the cell marker one I showed you was a really bright fluorophore, and then the hedgehog readout is a really dim fluorophore. So both seem to work, but I wonder about trying to compare them and if there might be issues there. So I'm hoping to find, like, there's all these new fluorophores like Apple and all these other ones that are supposed to be like brighter red fluorophores. So I'm hoping to engineer a fish that has more comparable fluorophores, and ideally on the same locus. That way it's less of a pain to get them all in the same fish.
Well, sounds wonderful. Well, if we don't have any more questions, I just want to say thank you to everyone. Thank you to our guest speaker, Dr. Everson. It's really wonderful having you here and sharing some of your research.
Yeah, thank you, everyone.
Alana, did you want to say final words? Sorry.
It's okay. I was just going to say thank you, Josh, for joining us. That was great. And be sure to join us next month. We'll send out a calendar invite for the next combo CATCHA Earth Center series. Happy holidays, everybody.
Thank you.
Bye.
Bye.